Good morning, students. Our today's topic is factors affecting enzyme activity. The activity of an enzyme can be affected by a change in the conditions which can alter the tertiary structure of the protein. These includes temperature, pH, change in substrate concentration, or binding of some specific chemical that regulates its activity. So first about temperature and pH. Enzymes generally function in a narrow range of temperature and pH. Each enzyme shows its highest activity at a particular temperature and pH called the optimum temperature and optimum pH. Activity declines both below and above the optimum value. Low temperature preserves the enzymes in a temporary inactive state, whereas high temperature destroys enzymatic activity because proteins are denatured by heat. So in the figure 9.7a, enzyme activity and pH graph is shown. At particular point at the top, at that pH the enzyme activity is maximum. This point is known as the optimum pH. At the optimum pH this activity is maximum, and below that also its activity decreases, and after that also its activity decreases. Similarly, for the temperature also, one optimum temperature is there at which the enzymatic activity is maximum. When temperature is low, the enzyme becomes inactive and its activity decreases. Similarly, at high temperature, the denaturation of protein occurs, that then also its activity decreases. In the graph with the concentration of the substrate on enzyme activity, that C diagram, here the velocity of the reaction with the substrate. Initially, as the amount of substrate increases, the velocity of reaction increases. But after some times it becomes static because now no more enzyme molecules are available for that substrate. So next topic is concentration of substrate. With the increase in substrate concentration, the velocity of the enzymatic reactions rises at first. The reaction ultimately reaches a maximum value which is Vmax, which is not exceeded by any further rise in concentration of the substrate. This is because the enzyme molecules are fewer than the substrate molecules, and after saturation of these molecules there are no free enzyme molecules to bind with the additional substrate molecules. एक लिमिट के बाद में सब एंजाइम्स मॉलिक्यूल के साथ में सबस्ट्रेट कंबाइन कर लेते हैं. अब और एंजाइम मॉलिक्यूल्स अवेलेबल नहीं हैं कि वो सबस्ट्रेट के साथ कंबाइन कर सकें, इसलिए एक सैचुरेशन स्टेज आ जाएगी. The activity of an enzyme is also sensitive to the presence of specific chemicals that bind to the enzyme. When the binding of chemical shuts off enzyme activity, the process is called inhibition and the chemical is called inhibitor. कई बार कुछ मॉलिक्यूल्स आकर एंजाइम्स की एक्टिव साइट में कंबाइन कर लेते हैं और उसकी रिएक्शन को भी रोक दे रहे हैं. ऐसे मॉलिक्यूल्स को हम इनहिबिटर्स कहते हैं और प्रोसेस को बोलते हैं इनहिबिशन. व्हेन द इनहिबिटर क्लोजली रिजेंबल्स द सबस्ट्रेट इन इट्स मॉलिक्यूलर स्ट्रक्चर एंड इनहिबिट्स द एक्टिविटी ऑफ द एंजाइम, इट इज़ नोन एज कम्पिटिटिव इनहिबिटर. अगर वो इनहिबिटर मॉलिक्यूल की स्ट्रक्चर सबस्ट्रेट से मिलती जुलती स्ट्रक्चर है तो वो आकर एंजाइम की एक्टिव साइट में कंबाइन कर लेता है और इसको हम बोलते हैं कम्पिटिटिव इनहिबिटर, क्योंकि ये मॉलिक्यूल रिएक्शन को स्लो कर रहा है. ड्यू टू इट्स क्लोजर स्ट्रक्चरल सिमिलैरिटी विद द सबस्ट्रेट, the inhibitor competes with the substrate for the substrate binding site of the enzyme. क्योंकि इस इनहिबिटर की स्ट्रक्चर सबस्ट्रेट से मिल रही है तो ये अपना कॉम्पीट करता है सबस्ट्रेट के साथ में ताकि वो जाकर उसकी एक्टिव साइट में कंबाइन कर जाए. Consequently, the substrate cannot bind and as a result, the enzyme action declines. For example, inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate succinate in structure. Such competitive inhibitors are often used in the control of bacterial pathogens. Classification and nomenclature of enzymes. Thousands of enzymes have been discovered, isolated, and studied. Most of these enzymes have been classified into different groups based on the type of reactions they catalyzed. Enzymes are divided into 6 classes, each with 4 to 13 subclasses and named according by 4-digit number. These are oxidoreductases or dehydrogenases reaction in which one molecule is reduced and one is oxidized. Then transferases, hydrolases. Hydrolases means in the presence of water. Lyases, here breakdown occurs, removal of group occurs from the substrate. Isomerases, some enzymes catalyzes the isomer reactions, that is conversion of one form to the another. Ligases, some enzymes help in joining the compounds. So just learn one this type of reactions and their names. Next is cofactors. Enzymes are composed of one or several polypeptide chains. However, there are a number of cases in which non-protein constituent called cofactor are bound to the enzyme to make the enzyme catalytically active. In these instances, the protein portion of the enzyme is called apoenzyme. 3 kinds of cofactors may be identified: prosthetic group, coenzyme, and metal ions. So first, enzyme can be of 2 types, which contain protein part and non-protein part. Protein part is known as apoenzyme and non-protein part is known as cofactor. This cofactor again can be of 3 types: prosthetic group, coenzyme, and metal ion. Prosthetic groups are the organic compounds that are tightly bound to the apoenzyme. Prosthetic group वो नॉन प्रोटीन पार्ट है जो कि प्रोटीन पार्ट के साथ में, दैट इज एपोएंजाइम के साथ में बहुत टाइटली फिक्स हो जाता है. फॉर एग्जाम्पल, इन peroxidase and catalase, which catalyze the breakdown of hydrogen peroxide to water and hydrogen, heme is the prosthetic group. You have to learn this name. Next is coenzyme. Coenzymes are also organic compounds, but their association with the apoenzyme is only transitory, that is for some time only, usually occurring during the course of catalysis. So coenzymes, these are the compound जो कि प्रोटीन के साथ में कंबाइन करते हैं फॉर कुछ टाइम के लिए ही, जबकि वो रिएक्शन को कैटलाइज करना होता है. फॉर एग्जाम्पल, coenzymes serve as a cofactor in a number of different enzyme catalyzed reactions. The essential chemical component of many coenzymes are vitamins. For example, coenzyme nicotinamide adenine dinucleotide and NADP contains the vitamin niacin. In the NAD and NADP reactions, vitamin niacin acts as a coenzyme. A number of enzymes require metal ions for their activity, which form coordination bonds with side chains at the active site, and at the same time form one or more coordination bonds with the substrate. So example, zinc is a cofactor. for the proteolytic enzyme carboxypeptidase. Catalytic activity is lost when the cofactor is removed from the enzyme, which testifies that they play a crucial role in the catalytic activity of the enzyme. With this, our chapter Biomolecule is complete. If you have any doubt, you can ask me. Thank you. So here, enzyme consists of two parts, protein part and non-protein part. Protein part is known as apoenzyme and non-protein part is known as cofactor. Cofactor again can be of three types: prosthetic group, coenzyme and metal ion. Prosthetic group tightly bound to apoenzyme, coenzyme transitively bound to apoenzyme, and metal ion, that is zinc, etc. they act as a cofactor. I hope this is clear.